For all of human history, we've treated aging as an inevitable law of physics, a one-way street of wear and tear where a biological clock ticks down inside every cell from the moment you're born. But in 2020, researchers at Harvard University managed to reverse that clock. By resetting the biological age of eye cells in old blind mice, they were able to restore the animal's sight. This work has already moved into human clinical trials. By 2025, data is showing that clearing specific types of aged cells from the human body yields measurable improvements in both physical and cognitive performance. These results point toward a new understanding. The physical deterioration we call aging is a biological process, and biology is something we are learning to rewrite. To understand the mechanism, we have to look at the information theory of aging. While we once blamed mutated DNA, we now know that the genome of a 90-year-old remains remarkably intact. The real change happens in the epigenome, the system of chemical marks that dictates which genes are turned on or off. Think of your DNA as a compact disc. The disc itself, the genetic data, remains perfectly intact. But the epigenome is the laser reader. Over time, that reader gets scratched. The original music is still there, but the cell begins playing the wrong information. This information loss leaves cells confused. They stop dividing and refuse to die cleanly, instead becoming senescent zombie cells that linger in your tissue, secreting signals that drive chronic inflammation. This cellular senescence and the resulting epigenetic drift are now recognized as two of the primary drivers among the 12 accepted hallmarks of aging. Because the original genetic code remains uncorrupted, aging is effectively an information access problem. The software is malfunctioning, suggesting it can be debugged and restored. Dr. David Sinclair at Harvard Medical School set out to prove that this biological clock could be actively run backward through epigenetic reprogramming. In 2020, his team induced rapid aging in mice and then injected a cocktail of Yamanaka factors, proteins that can enter a cell and systematically strip away the accumulated chemical debris from the DNA strand. Wiping these epigenetic scratches clean allowed the cells to return to a youthful state. In the case of the blind mice, their retinal cells were reset, fully restoring their vision. By proving that physical deterioration is a reversible software glitch, Sinclair successfully demonstrated the uncoupling of chronological time from biological state. The focus is now on translating these results to human patients, moving from basic mouse models to clinical evidence. This is the field of senolytics, drugs designed to selectively hunt and dissolve corrupted inflammatory zombie cells while leaving healthy tissue untouched. In 2025, human data from Phase 1 and 2 trials are showing positive signals for patients with Alzheimer's, osteoporosis, and kidney disease. Clearing this corrupted cellular data creates the biological environment necessary for tissue to regenerate and regain lost physical function. We are already seeing the extreme edge of this uncoupling in individuals like Brian Johnson, who has turned his own body into a comprehensive biological experiment. Through the Blueprint protocol, he tracks 111 biomarkers to monitor his rate of aging. At 47 years old, his measured biological age is roughly a decade younger. This points toward a broader mathematical threshold known as longevity escape velocity, or LEV, a concept first proposed by Aubrey de Grey. LEV is the tipping point where medical science increases your remaining life expectancy faster than chronological time passes. If a year of research adds more than a year of life, the biological clock effectively stops. In this scenario, chronological age becomes an obsolete metric for determining the limits of a human life. However, the cost of this technology remains a massive barrier. Full-scale longevity protocols currently require millions of dollars in annual spending. Because of this cost, life-extending research is becoming concentrated within billionaire-funded programs and exclusive technological hubs. AI-assisted drug discovery is accelerating these therapies for those with the capital to fund them, while the global majority remains tied to the traditional biological clock. If aging becomes curable, time itself becomes a commodity. The central question shifts from cell engineering to the economic power required to access the cure.